the nerve impulse that needs to be passed on from one neuron to the other neuron. So now what is happening is it reaches this end of the axon. Now, what is there in this end of the axon? As we have said, that this round things that you are able to see here, they are your vesicles. Sorry. These are your vesicles that you can see. Okay. Inside these vesicles, what you are having is your neurotransmitters. Now understand one thing, we will go this in details. So maybe one neuron would be able to produce only one type of neurotransmitter. So they are specific, like which are the neurons which are there. So you can have acetylcholinergic neurons and all these kind of things, okay? Because the receptors which should be there, that is also going to be specific for the neurotransmitter, okay? So when these vesicles come, so there are again, these vesicles are already present in the knob. Please understand that they are already present in the knob, okay? So this region which you see, which is very much closer to your synaptic cleft, okay? So that can be the active region, okay? That means the vesicles which are present in this region, they are ready to get your, to get fused with the membranes of your axon and then get released in the synaptic cleft. So now you say that active hai ka kya matlab hai? That means these vesicles are ready. Now, when can these vesicles be ready? Because on their surfaces, they require some proteins, okay? On these vesicles require some proteins so that they can get fused like this in the membrane. So these vesicles will obtain those surface proteins which are there, whereas these vesicles which are not near the active region, they will not have those specific proteins which are required for the fusion, okay? So now when these nerve impulse arise, okay, at the end of the axon, there are again few chemical changes that takes place that will give the signal to these vesicles to fuse. Otherwise, they will not fuse. Until and unless you have an impulse coming from the cell body, these vesicles will not fuse. Okay, so there are they are usually calcium dependent kind of your change in the chemical and your electrical changes that takes place at the end of your these neurons. Okay, so now once these neurotransmitters have already been released, what happens? This is we have talked about, this region which we are talking here is your postsynaptic neuron. Postsynaptic neuron means it is the other neuron, and which is the receiver part? It is your dendrite. Dendrite is the one which is going to receive the signals, right? So on the surface of the dendrite, on the membrane of your dendrons, what you will have is these kind of your receptors, okay? They are your receptors, okay? Now, most of the time what happens is these receptors are bound to, or they are conjugated with channels. What they are conjugated with? Channels. What are channels? You have learned earlier. What are channels? They are like, they are regions, or you can say they are the way for your ions to move from your one place to the other. So membranes, we know they are semi-permeable in nature, and mostly the ions, okay, which are charged ions, they require specific channels to pass through this membrane. They cannot easily pass through the membranes on their own. They cannot diffuse, okay, they cannot diffuse on their own. So these receptors are associated with these channels. So what happens? So they are your receptor associated channels. So whenever your neurotransmitter comes and binds, you can see over here, if you closely look, sorry, if you have a closer look to your whatever your picture is there, you can see that wherever, so this is the three receptors where your channel or where your neurotransmitter has already bound. So these receptors are open. they have opened these channels. Okay, they have opened these channels for the ions to come inside, whereas these receptors, they have not yet bound to your neurotransmitters, and hence they are closed. Okay, so these channels play a very important role in changing the potential. I have told you that these are your electrical impulse. So when your electrical impulse is there, definitely it is related to your potential difference. Okay, it would be related to the potential difference. So what happens, or how this potential difference is going to be created? Let us understand that. Okay, so you have already learned about what is the feature of your having different concentration of ions across your across different types of your membranes, right? So here we know that the resting potential of any membrane is -70. The resting potential of any membrane is -70. So what does that mean? What does that mean? It is -70 MV millivolts. Okay. So what does that mean? It does not mean that there are more of negative ions inside and it is more of positive ions outside. No, that is not the case. Here what happens is, if this is your cell, okay, this is your cell, there would be less number of positive ions inside and there would be more number of positive ions outside. Okay, there would be more number of your positive ions outside. So when you have that difference, so bahar bahut zyada positive ions hai और अंदर बहुत कम positive ions. And which are the positive ions which are more outside? You have your sodium plus ions are more. You have your potassium plus ions which are more. You have your calcium plus ions which are more outside the cell. Okay, outside the cell. So that is how the resting potential becomes -70. मतलब अंदर बहुत कम positive ions है और बाहर बहुत ज़्यादा positive ions है. So it creates a gradient. If you remember that last time in your S5 you have learned about your sodium potassium pumps, like how your 2 potassium ions will come inside and 3 sodium ions are going outside. So that again changes the potential, like you are putting more sodium ions outside, like more charges are going outside and less charges going coming inside. So that also creates a gradient. Less positive charges are inside and more positive charges are outside. So because of this gradient, now what happens when these channels open up? Because of the binding of these neurotransmitter, what happens is these channels will open up. So when these channels open up, the sodium plus ions come inside. Okay, the first ion which usually comes inside is your sodium plus ions. Okay, sodium plus and your potassium ions, they come inside. So what happens if they come inside? So this is called as minus. Being minus is polarized. Okay, so your membrane is polarized in nature. It has some charge, right? So now when your sodium ion starts coming in, that means there is inpour of positive ions. So samjho, now 20 ions have come inside. So what will happen? -70 and you have +20. So what is the answer that you will get when you are calculating this? You are getting -50. Now more amount of your +20 came inside, more amount of your 20 sodium ions came inside. Again, -50 +20, so that will become -30. So what is happening if you consider a number line? Okay, you have positive charge over here, you have negative charge towards here, and you have 0 over here, right? So here is say minus 70. So as it is coming towards, you are coming minus 50, minus 30, that means it is coming towards 0. So that means what is happening is it is becoming depolarized. So because of the binding of your neurotransmitters, okay, because of the binding of your neurotransmitters, what is going to happen is the ion channels are going to open up, okay? So because of the opening of your ion channels, these potassium and sodium ions will rush inside the dendrites, or it will come inside the other neuron which is there, and it will lead to the depolarization of your neuron. Okay, this depolarization is going to be passed on. Okay, so what happens is, abhi yahan pe, this is your receptors are present only at this particular region of the postsynaptic neuron. Okay, सिर्फ यहाँ पे जो postsynaptic zone है वहीं पे present होगा, but you have your entire so for example समझो यहाँ से पूरा तुम्हारा neurons है, right? You will have the entire neuron. Now there would not be any kind of your receptors at this region, but here you will have another channels which would be your voltage specific channels. Voltage specific channels matlab kya? They will open up whenever there is a change in the voltage. Okay, they will open up whenever there is your change in the voltage sensitive bolte hai usko, voltage sensitive channels. Okay, so when your sodium ions are coming in and when the depolarization occurs at this particular region, this depolarization would be sensed. Okay, it would be sensed by these whatever your voltage sensitive channels are there, and these voltage sensitive channels will also open up. Okay, and they will have, so jab ye channels open up honge और भी sodium ions और भी potassium ions अंदर आएंगे. Okay, more amount of your positive ions will come inside. So that is how. So now slowly it is going to, so as if I say over here that initially your whatever your receptors were there that got bound to here, but now slowly that depolarization travels from this dendrite. Okay, so all this entire cell, all this entire neuron will undergo change in the polarization, or you can say undergo change in the voltage. Okay, the more and more sodium ions and potassium ions will come inside this particular neuron whenever there is change in the dendrites that are taking place. And finally, it is going to reach to this axon end. And again, from this axon end, neurotransmitters are going to be released that will further make this change in the whatever the next neuron that is there. Am I very much clear, students? Understood what is happening? Okay, so that is what I was talking about. कि यहाँ पर you are having your neurotransmitters that get released because of your binding. Okay, these neurotransmitters will go and bind to these receptors. Because of this binding to the receptors, what is going to happen is there are many channels that will open up. Okay, so here तुम्हारे channels है. Okay, so these are the channels which are there. Okay, so which are receptor associated. That means when your neurotransmitter comes and binds, okay, when your neurotransmitter comes and binds, there is change. There is change in the channels. They will open up and allow the ions which are there in this region. यहाँ पे जो region है outside the cell, I told you outside the cell the sodium ion concentration, potassium ion concentration is always high. So they will rush inside because there is a gradient. It will move from your higher concentration to your lower concentration. They will rush inside. So क्या हो जाएगा ये region में, this region, what happens is there is change in the voltage. There is a depolarization. So when you have minus 70, now it is moving towards 0. depolarization will take place. Now you can see these are other type of proteins, these are other type of channels which are present on the other regions. Now these channels are like voltage sensitive channel. तो यहाँ पे जो change आया, those changes are going to be understood by your these voltage sensitive channels. They will further open up and they will allow more and more ions to come inside. That is how the electric axis will pass from one, it will pass from, transmit from one neuron to the other neuron. Am I clear? Students, have you understood till this much?
Ma'am.
Okay. Anybody who wants me to repeat this, kindly type repeat.
Ma'am, can you please repeat the voltage sensitive channels, the last part?
Yeah, see, voltage sensitive channels kya hote hai ki whenever there is change in the voltage, so we saw that wherever you had these receptors associated channels, so wo region mein, so it is localized, kind of localized, because there are not so much number of channels which would be present at that particular region, right? So it is a little bit localized part. So ab just like for example, we were seeing this. So what happens is ye region, this region only, this change will take place, right? So I want the electric impulse should be strong, then only it will be passed on through this entire neuron. So that is the reason why such kind of voltage specific channels are also present on the entire neuron. So as the voltage changes in this region, now ये region में change हुआ, तो what will happen is this voltage sensitive channel is going to sense that change. Okay, that -70 था पहले, अभी now it is changing towards, it is getting depolarized. That change is being understood by these voltage sensitive channels and they will open up. Okay, when they open up, they will allow more and more ions, positive ions, to come inside. When they come inside, again this region will also get depolarized. So these channels which are there, they will sense that change in the voltage, and now they will open up and again allow more ions to come inside. So again this region will get depolarized, that will be sensed by these voltage sensitive channels, again they will open up, allow the more ions to come inside. Understood? So that is how this electric impulse will travel throughout the entire neuron. Am I clear, students? Everyone?
Yes, ma'am.
Okay. So, yes, Soumya, is it clear about the voltage sensitive channels?
Yes, ma'am.
Okay, thank you. So the transmitter passes across the 10 to 15 nanometer, typically 12 nanometer synaptic cleft. So what is the size? It is just 10 to 50 nanometer, and specifically you can talk about, most of the times it is only 20 nanometer is the width of your synaptic cleft between the two cells, and induces a change in the electrical potential of your postsynaptic membrane of the next neuron. That is what I have just explained you. See, one more thing is, what I have just explained you is a excitatory transmitter. Okay, there are two types of transmissions. Either a neuron could be excitatory in nature or it could be inhibitory in nature. Okay, we will go in depth of that. Okay, we will understand what is the role of inhibitory neurotransmitters also, but understand that there would be two types: excitatory neurotransmitter and inhibitory neurotransmitter. Inhibitory neurotransmitter का क्या role रहेगा? To inhibit the impulse. Okay, why this inhibition is required? See, inhibition is required for kind of fine tuning. See, for example, let me give you a very simple example. Samjho, I'm just taken a needle and I've just touched on your skin. Okay? And in a person, without even saying कि उसको दर्द हो रहा है कि नहीं, he just screams, बहुत दर्द हो रहा है, ऐसा करके scream करेंगे. तो how is our reaction? पागल है क्या? Why to give such a big reaction for when you did not have any kind of pain also? है ना? That is our attitude. Why? Because when there is no impulse, you should not give a reaction. There should not be a reaction, right? Whereas अगर condition उल्टा है, समझो एक person को बहुत जोर से मार लगता है और वो चिल्लाता भी नहीं है, या there is no reaction from that person. What do you say? Dumb है क्या? He doesn't understand also that he is getting hurt. Are you getting my point? What I mean to say, that our body functions in such a way, our brain, our neurons function in such a way that as the strength of the impulse increases, there is the reaction change भी उतना ही ज़्यादा होता है. अपना reaction is directly proportional to the impulse which is there. Am I right? जो तुम्हारा like sensory organs, whatever the sense you are getting, that sense अगर strong है तो तुम्हारा reaction भी strong होता है. So that control in your body is very much required. So even if, see, when I am putting that needle on your skin, it you can feel it, right? You can feel it. Why you can feel it? Because your neuron is taking that impulse. Your neurons on the skin is taking that impulse. But your brain is not giving any kind of a signal to you to give that reaction. Why it is not giving that signal? Because the impulse was not very strong. Why the impulse was not very strong? Because in that case, your inhibitory neurotransmitters were stronger as compared to your excitatory neurotransmitters. Okay, I'll discuss this in much detail in the next lecture, don't worry. But for now, just understand that for fine-tuning our entire system, it is important that you have your excitatory neurotransmitters and your inhibitory neurotransmitter. So excitatory neurotransmitter क्या करता है? Depolarization. I told you that when it is minus 70, it tries to bring towards zero. So it is going for depolarization. Inhibitory neurotransmitter does just the opposite. It will open up the channels. Yes, similarly as it has done for your excitatory, it will also open up channels. But here, in this case, it will open up channels for chloride ions. What is the charge that chloride ions have, students? Negative charge, right? It is Cl minus, right? So now, instead of positive charge, negative charge ions are coming inside. So what will happen? Minus 70 में अगर मैंने minus 30 add किया, what will happen? It will become minus 100. Agreed or not? It is becoming minus 100. That means it is becoming more polarized. Right? It becoming more polarized. That means it is giving a resistance for the passing on to the impulse. Okay, so that is the role of your inhibitory neurotransmitters. So that is what we were talking about, that excitatory neurotransmitters usually cause depolarization of the membrane, as I've already explained it to you. So by this, we mean that the membrane potential, which is a resting neuron, is minus 50 to minus 70 MV. It falls nearly zero. So often, as a consequence of an increased permeability to sodium ions and resultant inflow of this sodium ions. So the resulting post-synaptic potential, really a drop in the potential difference, is propagated to the cell body. That means it has to move through the cell body to the axon. It has to reach from that, whatever your, this thing, from your dendrites to the axon, and again it has to initiate an action potential. So this is a narrow spike-like region of depolarization that travels down. So Sara Pura, so we show that as if an electric change is taking place, an electric spike is running towards a neuron. So this is what is running? It is your depolarization that travels down. Okay, so that travels down at the axon at a constant velocity and with an undiminished intensity. मतलब उतना ही strong वो अंदर तक, like at the till the end it is going to travel. Okay, so this is the same thing that we have talked about. This is the whole thing. So you have the postsynaptic neuron where your receptors are there, and these receptors are associated with your channels. So when your neurotransmitter comes and binds, so what will happen is it is going to be released. So here, this another picture also shows you that how your action potential travels down the axon by one layer. And here you can see again some, as we were talking about the other day, that there is an interaction between the axon and your cell body also. So that is how your empty vesicle. So once the vesicle releases out, यहाँ पे जैसे तुम देख रहे हो कि this vesicles which are filled up with your neurotransmitters, so they are releasing the neurotransmitter in this cleft. Okay, so once this releases the neurotransmitter, it becomes empty. So those empty vesicles are again going to travel back to your cell body. It gets filled up with your neurotransmitters again, and again it will come back. So last time you had learned about microtubules. Yes, so these microtubules which are there, they are responsible for carrying these vesicles from the cell body to your end of your axon. Am I very much clear, students? Okay, just I'll show you one video for this entire potential. Just give me 1 minute.